Should we experiment on the human genome? Through research on the scientific and ethical lenses, I was led to the claim that further experimentation on the human genome should be encouraged, but should only be used for the prevention and curing of diseases. Before any ethical questions can be answered, it is essential to understand the science behind genetic editing so we can recognize the limits, risks, and potential of this technology. Several different methods of genetic editing have been developed, but CRISPR has become prevalent because it's faster, cheaper, more accurate, and more efficient than its alternatives. CRISPR, along with the Cas9 protein, is a genetic engineering tool that is able to cut out pieces of a person's DNA and replace those pieces with altered versions of that DNA, essentially allowing certain genetic traits to be changed at will. This technology was originally adapted from a naturally occurring genome editing process that occurs in bacteria. In this naturally occurring process, the bacteria first takes certain pieces of a virus's DNA and then uses those pieces to create segments of DNA called CRISPR arrays. These arrays then allow the bacteria to recognize those viruses and viruses similar to it. When a virus is recognized, the bacteria produces RNA segments from the CRISPR arrays that lock in on the virus's DNA. The bacteria then uses the Cas9 protein to cut the DNA apart, which in turn disables the virus. The CRISPR and Cas9 method that can be used on humans is similar to, but not identical to, the naturally occurring version. In the lab, scientists select the DNA arrays, making the guide RNA that binds to the Cas9 protein. Just like in the bacteria, the RNA is used to identify the DNA sequence that needs to be changed. And when the sequence is found, the Cas9 protein cuts off from that point, and from that point, researchers can either allow the cell to replace the cut off DNA by itself, or make changes to the DNA by filling the gap with a customized DNA sequence. While most geneticists agree that CRISPR could be effective in the human body, there is still a lot of debate on whether this approach is safe for humans. The main safety concern is off-target edits, which could cause unwanted genetic changes. As visualized in the diagram on the board, off-target edits happen when the wrong piece of DNA is edited, and is usually the result of many sites in the genome having the same DNA sequence. These types of mistakes can sometimes be inconsequential, but at other times lead to deadly diseases like cancer. These mistakes highlight the biggest challenge that CRISPR and the Cas9 protein face, which is the delivery of CRISPR into different sites in the body. CRISPR's inability to travel to all parts of the body is why we are not yet able to use this technology on brain cells, heart cells, or muscle cells. However, there are certain single gene disorders, such as sickle cell disease, that are currently being treated using CRISPR on a trial basis. While the sample size is relatively small, the results regarding sickle cell disease have been almost exclusively positive. This success is likely to lead to greater acceptance of this kind of treatment, and with this acceptance comes a whole set of ethical issues that must be addressed. One of the biggest concerns of the public is that of designer babies. Theoretically, parents would be able to influence certain traits like hair color, eye color, height, muscles, and even intelligence. If this kind of technology were to become a reality, it would be an ethical nightmare and would leave parents who could not afford to boost their kids' intelligence at a significant disadvantage. And while this idea is extremely concerning, the reality is that top gene editing scientists say intelligence is too complex to engineer, and that's a good thing. Even in the cases of physical traits like height or muscles, there are dozens of genes involved that decide those things, and it will likely be a long time before CRISPR technology has the ability to effectively ensure these traits. In conclusion, it is important that we do not squander the medical potential of this technology because of fear, but instead embrace it and learn more about it so it can be perfected and eventually prevent every genetic disease.
What is some of the information that you started with that guided your research?
So obviously we started in class with the basic scientific of how the CRISPR and Cas9 was actually, worked out. And so that sort of gave me a couple questions of why doesn't the CRISPR find the wrong part of the DNA, but actually the right sequence, the sequence that they're looking for in a different part. And so that sort of influenced some of the off-target editing research that I ended up doing.
Okay, thank you. What are some impacts on our community?
Could you repeat the?
What are some impacts of your research on the community, or your impacts of your conclusion on the community?
Hopefully, the research I did sort of convinces the community a little bit more, because right now what it's being used for is really not very harmful, and I think I had research that I didn't include that said 80% of people and scientists agree that it's not a good idea to enhance traits like intelligence or physical traits. So I think that some of the fear people have is irrational. So.
Okay, thank you.